Hello and welcome to one more episode on Biostatistics. Today we shall look into experimental design. Whenever we start experimental research, what is most important is to plan and design the experiment in order to get appropriate data and desirable results. Experimental design includes formulation of objectives, planning and designing of the experiment. Experimental design is established on three important factors. They are validity, reliability and replicability. Whenever we conduct the experiment, the experimental result that we get should be validated, should be reliable, should be replicated, should be repeated by somebody else. Even if somebody else conducts the same experiment, they also should get the similar results. These are the three fundamental requirements of an experimental design. The concepts and principles of experimental design were laid down by two great statisticians. One is Charles Peirce, the other one is Ronald Fisher. C.R. Rao and Raj Chandra Bose have also contributed a lot to experimental design. The three fundamental principles of experimental design are randomization, replication, and local control. Randomization is whenever we are selecting the sample should be selected in an unbiased manner, in a random manner. I've explained about different sampling techniques in some other video. And the second important principle of experimental design is replication. Whatever treatment that you give to a particular type of subject or a sample, it should be given for more than one. Minimum 6 samples should be given the same treatment. This is called replication. The average of that has to be taken. Double blind technique or triple blind technique is used when we are doing the sampling in a randomized manner. This is what is called randomization. Whenever, let us say, you are testing a drug on a population of people, so in this case, neither the patient knows that they are taking the drug, nor the scientist who is giving the drug or doctor who is giving the drug to the people knows the drug, what drug they are using. So neither the doctor knows nor the patient knows. So these two are actually blinded in order to avoid any kind of bias, any kind of experimental bias. So this is called double blind technique. The third important principle is local control. Suppose you grow some plants on an experimental basis on a soil. We only study the plant characters and also the supplements that are given to the plants. But normally what we do is we don't give that much of importance to the soil, because soil also plays an important role. So thus soil becomes the local control, and giving importance to soil or local control is another fundamental principle of experimental design. In experimental design, in order to achieve maximum randomization, there are two techniques are used. One is completely randomized design, in which treatments are assigned in a completely randomized way. The other method is randomized complete block design, where treatments are assigned in specific blocks. The investigator has to be very careful about false positives and false negatives, which come as a part of your experimental design and experimental research. Generally, quadrates and grad sets are used for sampling in ecological studies. Whenever we do sampling in ecological studies, rank abundance curve or Whittaker plot is used. Thank you very much for watching this video. Actually, I have only touched the tip of the iceberg as far as experimental design is concerned. It is a very large topic, and it has a tremendous scope, and it is very essential whenever we are conducting research. Thank you very much, and we will meet again in some other class with some other topic. Thank you very much.